there is a number that most people never think about until the day it frightens them. It shows up on a small glowing screen after a finger prick, or on a lab report tucked between other numbers a doctor reads faster than you can process. And for millions of people, that number keeps climbing for years before anyone explains why. Here is the question almost no one asks out loud. What if the problem was never simply too much sugar, but something happening quietly inside your cells long before sugar ever became an issue? What if the real story of blood sugar has almost nothing to do with willpower, and everything to do with a conversation your body has been failing to have with itself? This is not a video about cutting out carbohydrates. It is not a video about starving your way to a better lab result. It is about understanding what is actually happening beneath the surface of high blood sugar, why the standard advice so often falls short, and what the research actually shows about restoring the body's own ability to regulate itself. By the end, you will understand your blood sugar in a way a rushed 15-minute appointment rarely has time to explain. Let's start with the part almost everyone gets wrong. Blood sugar problems are usually described as a sugar problem, but sugar is only the messenger. The real story is about a hormone called insulin, and more specifically, about how well your cells are listening to it. Picture insulin as a key and your cells as locked doors. When you eat, glucose enters your bloodstream, and insulin is released to unlock the cells so that glucose can move out of the blood and into the tissue, where it becomes usable energy. In a well-functioning system, this happens smoothly, almost invisibly. The key turns, the door opens, sugar moves in, blood sugar levels settle back down. But for a large number of people, especially after years of certain dietary patterns, chronic stress, poor sleep, and inactivity, something changes at the level of the cell. The lock is stiff. Insulin still shows up, still tries to turn the key, but the door doesn't open as easily anymore. Scientists call this insulin resistance, and it is one of the most misunderstood terms in modern health conversations. It doesn't mean your body stopped making insulin. In many cases, it means your body is making more of it than ever, desperately trying to force a door that has become harder to open. Your pancreas, sensing that glucose isn't clearing from the blood the way it should, ramps insulin production even further. For a while, this compensation works. Blood sugar might look normal on paper, even though insulin levels behind the scenes are quietly climbing. This hidden stage can last for years, sometimes over a decade, without a single symptom. This is part of why blood sugar issues can feel like they arrive suddenly. In reality, the groundwork was often laid long before the diagnosis. And this is exactly why simply removing carbohydrates from a plate, while sometimes useful in the short term, doesn't address the deeper mechanical problem. If the lock itself is the issue, reducing how many times you knock on the door doesn't fix the lock. It just knocks often. So what actually restore that sensitivity? This is where nutritional science becomes genuinely fascinating, because certain foods appear to work directly on this cellular communication problem. Not by eliminating entire food groups, but by changing the pace, the chemistry, and the inflammatory environment surrounding your cells. Let's walk through what the evidence actually shows, one piece at a time, because the way these mechanisms connect to each other is where this story gets interesting. The first piece of the puzzle is speed. Specifically, the speed at which sugar enters your bloodstream after a meal. A rapid flood of glucose forces a rapid, high-volume release of insulin, which over time contributes to that receptor fatigue we just described. Foods rich in soluble fiber play a critical role here, because soluble fiber has the unusual property of turning into a thick gel when it meets liquid in your digestive tract. That gel physically slows the movement of food through your stomach and small intestine, which means glucose is released into your bloodstream gradually instead of all at once. Fenugreek seeds are one of the most studied examples of this. Multiple clinical studies, including trials published in nutrition and diabetes-focused journals, have found that fenugreek supplementation can meaningfully improve fasting blood glucose and post-meal glucose response, likely due to its high concentration of a specific soluble fiber called galactomannan, alongside an amino acid that appears to support more efficient insulin release. It is a humble, inexpensive spice isle seed carrying a surprisingly well-documented physiological effect. Chia seeds work on a similar gel-forming principle, but with an added layer. Once soaked, chia seeds swell into a viscous gel that not only slows digestion, but also appears to reduce the overall glycemic impact of a meal when eaten alongside carbohydrates. Interestingly, chia also contributes plant-based omega-3 fatty acids, which brings us to the second major piece of this puzzle: inflammation. Here is something that surprises most people the first time they hear it. Insulin resistance is not purely a sugar problem. It is, in large part, an inflammation problem. Chronic low-grade inflammation, the kind that doesn't cause pain or obvious symptoms but quietly persists in the background for years, appears to interfere directly with insulin signaling at the cellular level. Researchers have identified inflammatory molecules that can physically disrupt the pathway insulin uses to communicate with a cell, essentially adding static to the signal. This is one reason two people can eat similar diets and have very different blood sugar outcomes. Their underlying inflammatory environment may be entirely different. This is where fatty fish enters the picture, and it is worth pausing here, because the research on omega-3 fatty acids and metabolic health has grown substantially over the past 2 decades. Fish like salmon, mackerel, and sardines are rich in EPA and DHA, two long-chain omega-3 fats known for their anti-inflammatory properties. Multiple population studies have linked regular fatty fish consumption to improved markers of insulin sensitivity and more favorable metabolic profiles. The proposed mechanism is that these fats help stabilize cell membranes and reduce the inflammatory signaling that interferes with insulin's ability to do its job. Walnuts offer a plant-based version of this same benefit, supplying alpha-linolenic acid, a precursor omega-3, along with fiber and protein that further blunt the post-meal glucose spike. Now here is where the story becomes genuinely surprising, because the next mechanism involves an organ most people never think about when they think about blood sugar: the liver. Many people are told their fasting blood sugar, the reading taken first thing in the morning, before eating anything, should naturally be their lowest reading of the day. And yet a significant number of people find their highest number is the one they wake up to. This confuses people deeply, because logically, if you haven't eaten all night, where is that sugar even coming from? The answer lies in a process called hepatic glucose production. Your liver stores glucose as glycogen and releases it overnight to keep your brain and body supplied with energy while you sleep. In a well-regulated system, this release is modest and tightly controlled. But in insulin resistance, this overnight release can become excessive because the liver, like other tissues, can also become less responsive to insulin's signal to slow down glucose output. The result is a liver that behaves like a factory left running on overdrive through the night, quietly manufacturing more sugar than the body needs, so that people wake up to numbers that seem to contradict the fact that they haven't eaten in 10 or 12 hours. This is where broccoli sprouts become one of the more fascinating entries in this conversation. These are not the mature broccoli heads sold in the produce aisle, but the tiny 3-day-old sprouted version, which contains dramatically higher concentrations, in some analyses 50 to 100 times more, of a plant compound called sulforaphane. A study conducted by researchers at Lund University in Sweden examined the effect of broccoli sprout extract on people with type 2 diabetes and found measurable reductions in fasting blood glucose, with the most pronounced benefit observed in participants whose blood sugar had been hardest to control through standard treatment alone. The proposed mechanism involves sulforaphane's influence on liver gene expression related to glucose production, essentially helping to quiet that overnight glucose factory. It is a striking example of how a food most people walk past in the grocery store may be doing more metabolic work than foods marketed specifically as diabetic-friendly. Let's talk about fat for a moment, because few nutrients have been more misunderstood in the context of blood sugar than fat itself. For decades, dietary fat was treated as the primary villain in metabolic disease, which led entire generations toward low-fat, high-refined carbohydrate alternatives that ironically may have worsened blood sugar control for many people. Avocados offer a useful case study in why this blanket fear of fat was oversimplified. The fat in an avocado is predominantly monounsaturated, the same category found in olive oil, and research suggests that replacing refined carbohydrates or saturated fats with monounsaturated fat can improve markers of insulin sensitivity. Beyond the fat itself, the fiber content in avocado slows gastric emptying, which again reduces the speed and size of the glucose spike that follows a meal. When paired with a carbohydrate-containing food, avocado appears to soften what would otherwise be a sharper rise in blood sugar. Eggs tell a related but distinct story, one connected to a phenomenon researchers call the second meal effect. This refers to the observation that what you eat at one meal can influence your blood sugar response at the next meal hours later. Because eggs contain essentially no carbohydrate and a strong combination of protein and fat, starting the day with eggs rather than a carbohydrate-heavy breakfast, like cereal, appears to produce more stable glucose readings not just immediately after eating, but through the following meal as well. This is part of why breakfast composition tends to have an outsized influence on blood sugar patterns across the entire day, more than many people realize. Cinnamon deserves particular attention, not because it is a miracle spice, but because the distinction between its two common varieties is rarely explained clearly. Most cinnamon sold in grocery stores is cassia cinnamon, which contains a compound called coumarin that in high daily doses over long periods has raised concern for liver health in some research. Ceylon cinnamon, sometimes labeled true cinnamon, contains dramatically lower coumarin levels and is generally considered the more suitable option for regular daily use. Research from human nutrition programs, including work conducted through USDA-affiliated research, has found that modest daily cinnamon intake, well under a full teaspoon, was associated with measurable improvements in fasting glucose among adults with type 2 diabetes. The proposed mechanism involves specific polyphenol compounds in cinnamon that appear to enhance how tightly insulin receptors bind glucose, alongside a mild slowing effect on gastric emptying, similar to what we discussed with fiber-rich foods. Apple cider vinegar is perhaps the most immediately measurable intervention on this list, and it is worth explaining why. The active compound is acetic acid, and research led by scientists including Dr. Carol Johnston at Arizona State University has demonstrated that consuming a modest amount of vinegar, roughly 1 tablespoon diluted in water, before a carbohydrate-containing meal can reduce the resulting blood sugar spike by a meaningful margin in people with insulin resistance. Acetic acid appears to work through two mechanisms simultaneously, slowing gastric emptying and enhancing glucose uptake into muscle tissue. This is precisely the kind of finding that surprises people, because something as simple and inexpensive as diluted vinegar is producing an effect size that begins to approach what some medications are designed to do, though it should never be as a replacement for prescribed treatment. And anyone on blood sugar medication should coordinate any dietary changes like this with their physician, since combining the two can occasionally lower blood sugar more than expected. And then there are lentils, which make an important and somewhat counterintuitive point. Lentils are, technically, a carbohydrate, and yet they are one of the most reliable foods for improving blood sugar stability precisely because of how their carbohydrate is packaged. Lentils combine resistant starch, soluble fiber, and plant protein in a way that dramatically slows glucose release compared to refined carbohydrates like white rice or white potato. This illustrates something important about the entire conversation around blood sugar. It was never really about carbohydrates as a category. It is about the structure, fiber content, and nutrient pairing of the food carrying that carbohydrate. A cup of lentils and a cup of white rice may contain similar amounts of carbohydrate on paper, but they produce very different physiological responses because one arrives at your bloodstream gradually and the other arrives in a rush. Taken together, these mechanisms describe three overlapping strategies your body can use to restore healthier blood sugar regulation. The first is simply slowing down how quickly sugar enters the bloodstream after eating, which reduces the demand placed on insulin in any single moment. The second is reducing the chronic inflammation that interferes with how well cells listen to insulin's signal in the first place. The third is addressing the liver's overnight glucose output, which is often the hidden reason fasting numbers stay stubbornly high even when someone feels they are doing everything right during the day. None of these strategies require eliminating entire food groups. They require adding foods and habits that support the systems already responsible for regulation. There is also a psychological dimension to this that deserves honest attention, because willpower-based advice tends to quietly set people up to feel like personal failures when blood sugar issues persist despite genuine effort. When someone is told their only path forward is restriction, and that path doesn't fully work, it is easy to internalize that as a character flaw rather than recognizing it as an incomplete strategy. Understanding the biological mechanism, insulin resistance, as a cellular communication issue rather than a moral failing around sugar consumption, tends to change how people relate to their own health. It shifts the emotional framing from guilt to curiosity, and curiosity is a far more sustainable motivator than shame ever is. This matters clinically too, because sustained behavior change is more likely when someone understands the why behind a habit rather than following a rule they don't fully believe in. It's worth pausing on how gradual and undramatic real physiological change tends to look in practice. Blood sugar regulation is not something that flips from broken to fixed overnight. Fasting glucose readings often begin shifting within 1 to 2 weeks of consistent changes, but deeper markers like A1C, which reflects your average blood sugar over roughly 3 months, take correspondingly longer to reflect meaningful improvement. This is important context because people frequently abandon promising changes simply because the timeline of visible results doesn't match their expectations. The body's feedback loop is slower than most modern habits are designed to reward, and understanding that mismatch in advance can be the difference between sticking with a change long enough to see it work or quitting 3 days before the data would have started to show it. Sleep and stress deserve mention here too, even briefly, because they are inseparable from this conversation even though they rarely appear on lists of foods for blood sugar. Poor sleep has been shown in multiple studies to reduce insulin sensitivity within as little as a single night of restricted sleep. Chronic stress elevates cortisol, a hormone that, among its many roles, signals the liver to release more glucose, working directly against the overnight regulation we discussed earlier. This means that even a perfect diet operating within a context of chronic sleep deprivation or unmanaged stress is fighting an uphill battle. The foods described here are powerful tools, but they operate within a larger physiological system that also includes rest, movement, and stress regulation, and none of these pieces work in complete isolation from the others. It's also worth being honest about what this information is not. It is not a replacement for medical care, and it is not a claim that food alone reverses every case of insulin resistance or type 2 diabetes for every person. Genetics, disease duration, medication status, and individual physiology all influence outcomes, and anyone managing diagnosed blood sugar issues, particularly those on insulin or other glucose-lowering medications, should make dietary changes in coordination with their healthcare provider, since improving insulin sensitivity can sometimes mean an existing medication dose becomes too strong for the body's new baseline. Responsible, informed collaboration with a doctor tends to produce far better long-term outcomes than either extreme, ignoring medical guidance entirely, or assuming medication is the only lever available. What makes this topic worth sitting with isn't just the mechanics of glucose and insulin. It's the broader idea underneath it, that many of the numbers we're handed in a lab report are not fixed sentences, but reflections of an ongoing conversation between what we eat, how we live, and how our cells respond. That can shift, not through deprivation and not through fear, but through a more precise understanding of what your body is actually asking for. The people who tend to see the most meaningful, lasting change aren't usually the ones who eliminated the most foods. They're the ones who added the right ones, consistently, and gave their body enough time to respond in its own language. So the next time that number on the screen or the lab report catches your attention, it might be worth asking a different question than the one you've been asking. Not what do I need to cut out, but what is my body still waiting for me to give it? That single shift in framing, from restriction to restoration, may be the most important insight in this entire conversation, and it's one worth carrying with you long after this video ends.